All right, hi everyone. My name is Maggie French, and I'm an assistant professor at the University of Utah. I'm also adjunct faculty at Johns Hopkins, and today I'm going to be talking about work that I did while I was at Hopkins full-time that's related to ensuring data quality when you're conducting research that uses data from the medical record. Brief outline of what we'll talk about. First, I just want to establish a foundation of why I'm particularly interested in using electronic medical record and why I'm therefore interested in the quality of that data, which is specifically due to my interest in precision rehabilitation. I'll then talk about a framework that we can use to think about data quality related to EHR data. I'll then also spend the bulk of the talk talking through some specific examples of how we can apply this framework to data from the electronic health record. And then lastly, I'll wrap up by talking about how we can apply data science principles to streamline data quality checks and to facilitate repeatability and reproducibility of research that we do that uses electronic health record data. So I'm particularly interested in how we can move neurorehabilitation towards precision rehabilitation. Precision rehabilitation is this idea that if we understand patient variability, we can move away from this one-size-fits-all approach and move to a more tailored, made-to-measure approach where we're providing more targeted care so that the right patient receives the right care at the right time. And the outcome of this is more effective care, meaning we get better patient outcomes, but it's also a healthcare system that's more efficient. So together, precision rehabilitation really moves us towards value-based care. So I want to highlight why electronic health records are important for precision rehabilitation. And to do that, I just want to boil precision rehabilitation down to three primary steps. The first step is that we need to understand patient variability, which requires that we curate heterogeneous data sets that have accurate measures of function. We then have to be able to predict that variability, meaning we have to use those heterogeneous data sets to help us build predictive models. And then we have to act on our patient variability during patient care. And what this means is that we then have to take those predictive models and we have to use them to actually guide clinical decisions. And so these three requirements of precision rehabilitation provides a really nice foundation for us to take advantage of electronic health record so that we can move towards precision rehabilitation. So at Hopkins, what we've done to move towards this is develop something called the Rehabilitation Data Repository. This repository is aimed to improve the access that researchers have to electronic health records so that we can make that shift towards precision rehabilitation. Within this data repository, we currently have over 18,000 individuals with stroke for whom we have their electronic health record data pulled into the data lake. The categories of data from the electronic health record can really be categorized into some of the examples you see here on the left side of your screen. So for example, we have rehab specific measures. So if you're a PT, you can think about this being things like gait speed. If you're an OT, maybe you're thinking about something like box and blocks. And if you're a speech therapist, maybe something like R-bands. We also have information about when people make an encounter with our healthcare system. So any visit they have, whether it's to a primary care physician, a neurologist, an orthopedist, a PT, OT, speech therapist, we have information about that encounter. We also have information about a couple other categories that you see here, but the real take-home point is that this data repository serves as a way for us to access a large amount of electronic health records. To give you a sense of how much data we have in this repository, I want to show you kind of the structure of that data. So this data is stored for us in a SQL server. What I'm showing here is a screenshot from that SQL database. A SQL server or a SQL database is a storage system where all of the data is related to one another. So in this example that we're talking about with our data repository, we have 64 different tables that contain data that have a relationship to one another. In our particular repository, the number of observations in each of these tables range anywhere from over 18,000 to over 4 million observations. And then within each of these tables, we have a number of different column names, which you can see a screenshot of here, And these can be thought of different variables that live within each of the tables. For each of these tables, we have between 12 and 48 different variables. And so you can see that we're talking about really large data when we're talking about working with this electronic health record here. The way we have our process set up is that this data is stored within a data lake or a data repository. And then when researchers want to use it to answer specific questions, they submit IRB protocols that allow them to access a subset of that data. Now, understanding data quality is important both at this stage where we have our data within our data repository and during each of these individual research protocols. And so what I'm going to talk about today is specifically how we can ensure that we have high quality data from the electronic health record that we can use in research. So what we're going to focus on today is data quality, specifically related to the electronic health record, because it's essential to us being able to draw valid conclusions from our data. A lot of people may be tempted to think that when we have large amounts of data, like I just described, that we don't necessarily have to worry as much about data quality because big data maybe is magical and can fix all of our problems. But the old adage of garbage in and garbage out, it holds even when you're working with this large amount of data. And it presents even more challenges because it's so much data to check the quality of. So there's a number of different frameworks that we can use to assess data quality. I'm going to focus specifically on one that was published by Kahn et al. In 2016. And in this framework, there's 3 categories of data quality. The first category is conformance, which is whether or not the values or the data within your database meets the structural constraints that you expect from it. In other words, is the data in the form that you think it's supposed to be in? The second category is completeness. What we mean by completeness is if the frequency of the data within your database is what you expect. The third category then is plausibility. And I think this is the category that most of us think about when we think about data quality. And this is really looking at if the values of your specific variable of interest are actually believable. Each of these 3 categories then have 2 kind of components to them. The first is verification, which means that you're assessing these 3 categories of data quality in reference to some sort of internal standard. So with the database that I just showed you, we'd be thinking about what we expect from the healthcare system of Johns Hopkins. Whereas for validation, you're looking at things in relation to external standards. And we'll go through some examples in just a moment that kind of distinguish between this verification and validation. So like I said, we're going to spend a lot of the talk today looking through some different examples of how we screen our data from this repository for data quality. And again, I'll use this framework to do that. So the first example that I want to go through is related to the patient identifiers. So this data set, again, this is our data repository here that I'm showing you. It has numerous tables, but I want to focus here on 2 tables in particular. One table, called the EPIC patient table, includes information about each individual patient, so there's 18,000 individuals with stroke. Another table is called the all encounters table. And what we would expect is that any patient who's in our repository should have some sort of encounter with our healthcare system. Otherwise, why would they be in our repository? And so this is a really like basic screen of data quality that I think is often overlooked, but it's an important one. And when we check this in our database at our repository, we actually see that there's 102 individuals who are in our patient table but don't have any encounters with our healthcare system. And what this really means is that we have to go back in and we need to inspect what's going on with these individuals to see if there's something wrong with our data set or if there's something else going on that's causing this issue in conformance. And this is an example of conformance because we expect the people who are in our repository to have an observation within that encounter table. Because they don't, it indicates that there's some sort of issue with the structure or the format of the data. The next example is related to sex. At Johns Hopkins, in our data, we have kind of two different variables that indicate individual sex. The first one is here where we have the sex actually spelled out, so female, male, and then we have an abbreviation of F, M, or U for unknown. When we think about conformance here, it's important that we understand what Hopkins expects for this field. And in fact, at Johns Hopkins, there are 3 different options that can be entered here, female, male, and unknown. So this demonstrates good conformance internally with what we expect in the Hopkins system. We can then think about completeness. We don't have anybody who sex is NA per se, so we have something documented for all of these folks. You could make the argument that because we have 2 people who the sex is unknown, that maybe there's some sort of issue with completeness there. But on the whole, most of our individuals have this variable. And then finally, when we think about plausibility, we can look at the percent here in this last column of the table. And we can check this against external standards. In this case, I've pulled data from the Maryland census to see the breakdown of gender within our state that Johns Hopkins serves. And you can see that on a state level, we have 51% female, and in our repository, we have 50.5% that are female. And so this demonstrates good plausibility in reference to that external source. The next example I want to walk through is related to race. So here I'm showing a breakdown of all of the races within our repository. When we think about conformance, again, it's important that we understand what is the standards at Hopkins. And in fact, these variables here are options that are available within our healthcare system. And so we have good internal conformance. However, these are not the categories that the US Census uses. So what I'm showing here are the 5 categories of race that the US Census uses. And you see that those clearly don't match what we have in our medical record at Hopkins. And so this is demonstrating that in comparison to an external reference, our data actually doesn't have great conformance. And so this is kind of what we would call maybe poor validation of this data related to the US Census. So I wanted to talk about plausibility with this data as well. And so what I've done to this data is I've actually collapsed all of these options for race so that they map on to the race options that the US Census Bureau uses. And when we do this, we can see that this is the breakdown of percentage for each of those races within our repository. To compare this, to understand the plausibility of this, we can compare this again to the census data. So what I'm showing in this plot here on the left is the census data for the state of Maryland. And you can see that the percentage is fairly similar when you look at our repository data and then the statewide data, indicating good plausibility compared to that external data source. So I now want to talk about some examples of data quality screens that we do related to outpatient physical therapy visits based on department. So related to conformance, at Johns Hopkins, we know that we have 4 outpatient physical therapy departments that see a vast majority of our individuals with stroke. And so the first thing we can do to check conformance is see if we have data from those 4 rehab clinics. And in fact, in our repository, we do, demonstrating good conformance related to what we expect the structure of the data to be. To examine completeness, we can look at the number of visits that occur within each of these clinic settings. And when we do this, we actually see a problem. We see that clinic 2, 3, and 4 here have visits, but clinic 1 and 2 have very few. For clinic 1, so much so that it actually doesn't even show up on the y-axis scale here. And so this is really demonstrating an issue with completeness. And we know that this is inaccurate because we know what actually is going on within our healthcare system. So this required us to do some digging. And what we found is that there's actually some outpatient visits that occur in a hospital setting or in regulated space that end up getting labeled as inpatient visits within our healthcare system. And so what I'm showing here is those missing visits that we had to go back in and find. And we were really only able to find these visits because we understood what was going on within our healthcare system. Related to plausibility, we can look at the frequency of these events over time. So now we're looking on the x-axis here at time. And interestingly, and so one way to do this is you can cross-reference with your operations team to see if this makes sense with what they know about their clinic census. And that's a really important thing to do because the operations teams, are in the weeds with what's going on within their clinic. The other thing you can do though is you can compare this to an external events. In this case, we see in all three of these clinics a decline in visits that occur at the beginning of 2020. And this makes a lot of sense given what we know happened to the world during 2020, the COVID pandemic, where we had a stark reduction across the healthcare systems of outpatient visits. And so this data does demonstrate good plausibility, both compared to our internal validity verification checks with our operations teams and with external sources. Last couple of examples I want to give are specific to rehab measures. So what I'm going to show here is some information about each of these rehab specific measures. When we talk about conformance here, we can focus specifically on the type of data that we have. For each of these measures, we are expecting to have some sort of numeric value. But when we check that within our data, we actually see that for 9-hole peg test, over 50% of our data is not entered as a numeric value, indicating poor conformance with that data. So to show you guys what this actually looks like in practice, I've pulled 10 random observations from the 9-hole peg test, and I'm showing that here. And what you can see is we have one random example here that is actually numeric, but then we have a lot of other observations where people are maybe adding the units, so seconds in this last example here. And then we have some examples where clinicians are making some note about the performance. And so this is actually really problematic when we want to use this data on the back end. Because we have poor conformance, we really can't actually use this data because it's not in the form that we expect. And if you skipped this step of checking the conformance of this data, you would move forward expecting there to be numeric values for this particular measurement, which in fact isn't the case. Related to completeness, another kind of summary stat that I've pulled here is how many of these observations don't have a score associated with them. So it's rare, but sometimes we have an observation of gait speed or the 9-hole peg test or one of these AMPAC scores, but we don't actually have a value for it. And so here for completeness, we're checking how often that occurs. Lastly, we'll look at plausibility of a couple of these rehab specific measures. The first one that we'll look at is related to gait speed. So this figure here is showing all of the values for gait speed from one field within our medical record. And if you're a PT, the y-axis here should jump out at you. Average gait speed for a healthy individual is about 1.2 meters per second. But here we're seeing observations as high as 40 meters per second. And so there's definitely something wrong with the plausibility of this data. What's probably happening here is that we have clinicians entering gait speed, which is represented by this chunk here. And then we have other clinicians that are actually entering the time it took patients to walk 10 meters. And so this is really problematic because we have two different units on the same data field. How we decided to address this was to actually make modifications to our electronic health record. And so here, what I'm showing is just a screenshot of what we built within our Epic medical record system, where we now have a field that specifically says that clinicians are to enter speed, time, so in seconds. And then in this field, it's expecting to have speed, so meters per second. And when we pull this new data field, we can look at the data quality and we see that it's drastically improved. We no longer have observations as high as 40 meters per second, and a vast majority of our observations are down where we would expect around 1 meter per second. And so this demonstrates that the change we made within our medical record system really improved the plausibility of the data that we got from the medical record, improving the data quality. The last example that I'll go through is related to the AMPAC mobility measurements. We use two different measurements of this within our healthcare system. The one here on the left is the inpatient AMPAC mobility, or the 6 clicks. And the one here on the right is the outpatient AMPAC short form. So when we think about the plausibility of the timing of these measurements, we'll first talk about the inpatient 6-click measurement. And interestingly, we see this kind of sharp increase in the number of observations around 2019. So you may think that this might be, there's something a little strange going on because of this change. And actually what happened in our healthcare system was that nurses started administering this measurement. And so even though this increase might seem a little suspect, we understand what's going on within our healthcare system, and we know that this observation is true, and therefore we have good plausibility based on the number of observations over the course of the data plotted here. Related to that outpatient measurement, we again see a dip in 2020. And again, when we think about what was going on in the world during that time, particularly the COVID-19 pandemic, this dip here makes a lot of sense, and again, demonstrates good plausibility. So I hope through these examples, you can see that checking the quality of the data doesn't just mean that you are looking at the value or the plausibility of the variable. You need to check and address the conformance, the completeness, as well as the plausibility so that we can actually trust the data that we're using. We also have to have a good understanding of what's going on in our healthcare system and what's going on in the world so that we can accurately assess that data quality. If you think about those dips in 2020, we might think that we were missing data if we forgot that the COVID-19 pandemic happened. And so having this understanding is really important. Now, these data quality checks can be quite consuming, but it really is essential. So much so that the record statement, which is one of the Strobe reporting documents that we're expected to use with this type of research, really highlights that data cleaning and processing, even though it's only like the very first step of the research and analytical pipeline, is really important, and we need to be reporting how we're doing it. And so the last thing I want to talk about is how we're streamlining this process of data quality checks to ensure rigorous and reproducible research with what we do at Hopkins. A key component of that is data dictionaries. And so I want to show you some examples of data dictionaries that we're using. Here, what I'm doing is I've shown a screenshot of one of our data dictionaries where we're looking at one table specifically, and we have each of these column names or variables listed in the data dictionary. We then have what their meaning is, what the expected values are, and then of any other important notes that we think are useful for future researchers. This is kind of a work in progress in our team because we have so many tables and so many different column names, but this really is important to ensuring that people can do these data quality checks in a timely fashion. We also have data dictionaries that dig deeper into specific tables. So what I'm showing here again is a screenshot of one of our data dictionaries that's specific to rehab measures. Our rehab measures are typically stored in this table here, and it has over 4 million observations in it. And so this data dictionary that I'm showing you a screenshot is essentially a map for where to find rehab specific measurements. And the third type of data dictionary that I want to show is a project specific data dictionary. Every project that we do has a data dictionary that follows this similar format where we specifically label the tables that we use for our data sources. We label ICD-10 codes that we use to develop our cohort, and then, et cetera, we include any other sort of essential information. What this approach allows us to do is to create shared and automatic code that we can use across our research team to streamline our process of data quality check. Our team specifically uses R, but you can certainly do this in things like Python and MATLAB as well. And then we're in the process of trying to develop a GitHub repository, which is an online platform that allows us to have version control with these shared codes. The snag point here is really that we need to make sure that we're not saving any sort of PHI data on GitHub. And so we're working on putting kind of checkpoints in place to ensure that when we're saving our version controlled codes onto GitHub, that we're not violating any sort of privacy policies. Now, this is great, but this is really only good for us internally at Hopkins. What we really need to be moving towards is using things called common data models. Common data models can be thought of as a standard language that can be used across settings. There's a couple of different frameworks for this. One is called PCORNet, another is called Sentinel, and then the last one here is called OMOP, and this is actually what we're moving to at Johns Hopkins. What this allows us to do is have a standard language that we can use at Hopkins and that anybody else in other healthcare systems can use. So to illustrate what OMOP allows us to do, we can talk about gait speed as an example. So at Johns Hopkins, gait speed is coded in our medical record with these two different codes. But OMOP, that standard language, has one value that is unique to gait speed, and that value is shown here. And so if we have this standard code from our common data model, or OMOP, we at Hopkins can refer to gait speed the same as folks at University of Utah, at Pitt, at Brooks Rehab Hospital, across numerous healthcare systems. And this would allow us to create code that we can use to screen our data quality at each of these healthcare systems, so that not everybody's having to go in and redo work that other healthcare systems have done. The other advantage of OMOP is that they also have shared tools that researchers can use. And a great example of this is their data quality assessment dashboard. This is a screenshot of it. And they use that same format or framework that we talked about today, where they have 3 categories and then 2 components of each category. And you can run this your data through this data quality dashboard locally so that it complies to your data security policies. And it can screen all of your data for its data quality. And so this is really a very helpful way for us to be able to streamline this process. Now, I do think it's important to mention that this isn't a magic bullet. All healthcare systems have nuances, and different research questions have different metrics that are of interest. But this type of community approach can really facilitate rehab research that uses this type of data and ensure the quality of the data that we're using. So in summary, electronic health records have a lot of potential for helping us conduct impactful research. I'm particularly interested in it as it pertains to precision rehabilitation. Data quality needs to be assessed for conformance, completeness, and plausibility. And we have to understand what's going on within our healthcare systems and within the world for us to accurately understand the quality of our data. Lastly, data science and reproducibility research principles are essential for conducting the research with electronic health records, and in particular for screening our data for data quality. These type of skills, though, are really challenging. And so I do want to provide you guys with a resource called Repo Rehab. This is a relatively new educational program that's out of USC, and they're really targeting trying to develop these data science skills within our rehab community. And so if you're interested in trying to improve your ability to automatically check your data for data quality, I recommend that you check out this website. Otherwise, thank you for your time. Please feel free to reach out to me with any questions or comments. My email address is listed there. And thank you guys for your time and for listening today.